Another great thank you to Lindsay for that informative presentation, and a big thanks to the audience. Some really, really great questions. So from here, I would like to welcome Sergey Rakilin to the stage. He is our Executive Director for our Critical Path for Sickle Cell Disease. So he's going to share a little bit with you on endpoint selection.
And you do. Okay. Hello, everyone. Can you hear me? It's a great honor to speak in front of such a wonderful audience. Thank you for having me here. The title of the presentation that I have today is Endpoint Selection for a Disease with Highly Heterogeneous Manifestations. And I'm an executive director of the Sickle Cell Disease Consortium. Our consortium is relatively young. We're only 3 years old, so we're still trying to catch up with other more advanced consortia in developing models. So I will briefly introduce sickle cell disease to you and outline potential solutions in identifying endpoints for that particular disease. So sickle cell disease affects approximately 100,000 Americans and more than 20 million people abroad, worldwide. And there are 4 therapeutic drugs that are currently on the market that partially alleviate the symptoms of the disease, making it a clear unmet medical need. As of 2023, there are 33 pharmaceutical companies that are pursuing and developing treatments for sickle cell disease, and the number of companies developing small molecule drugs versus genetic therapy approaches approximately split in half. Half of the companies are developing gene therapy, making it a forefront of the development of those treatments that would be applicable for other rare diseases. So the points of today's presentation is to kind of give sickle cell disease presented in a form of the example of the identifying endpoints and identifying treatments that will be applicable to other rare diseases as well. What's important on that particular slide is that there are 2 BLAs that are currently being reviewed by the FDA. And we hope that sickle cell disease will be one of the first diseases that could be cured by the gene therapy approach. Now, I will show you the point of the next slide is a simplified version of the molecular mechanism of the sickle cell disease. What you can see on the left upper part of the slide is that a point mutation in the hemoglobin gene causes rather decreases its ability to bind oxygen. As a result of that, hemoglobin polymerizes in lengthy fibrils, which become so long that it stretches the generally round red blood cells into sickle cells. And at some point, it causes hemolysis. Those cells break apart. This is a very important process because the half-life of the sickle cells is approximately 10 days compared to 40 days of the red blood cells in general, normal red blood cells. Now, as a result of that hemolysis, the occlusion of blood vessels occur. And that occlusion could happen anywhere in the body. It could happen in your hand, it could happen in your leg, it could happen in your brain. And that occlusion correlates with a great deal of pain that patients experience. Now, in the next slide, summarizes heterogeneous manifestations of the sickle cell disease that affect different organs. And you can think of that virtually every organ related to the penetrated blood vessels is affected. You can think of nervous system, and manifestation would be pain or stroke. You can think of cardiovascular system, where the acute chest syndrome will be prevalent. You can think of the reticular endothelial system where anemia or hemolysis could occur. Musculoskeletal system could be equally affected in a form of like ulceration. You can think of urogenital, gastrointestinal system, many other systems within human body that are affected by that disease. So this is a perfect example of the heterogeneous disease with heterogeneous manifestations that affect virtually every organ of the human body. Now get yourself in the shoes of the chief science officer or chief clinical medical officer of the pharmaceutical company. Try to come up with the clinical endpoints in order to develop a new therapeutical treatment for that particular disease. There are several options there. I divided those options into two groups. The first group relates to vaso-occlusive crisis, and they describe different aspects of that part. And the second group, I will talk about that a bit later, relate to surrogate endpoints or functional endpoints that are not directly related to vaso-occlusive crisis, but they're directly related to the mechanism of action of the drug itself. Let's start with the vaso-occlusive crisis-related endpoints. The first one, the most obvious one that we mentioned in the previous slide, is pain. The problem with the pain is that A, it's highly subjective. B, there was an observational study, what is called PACES, that was completed in 2008 and was completed by a distinguished professor, Wallace Smith, who specifically looked at the distribution of the pain within those patients where it occurred. And he, in this particular diagram that he summarized as a result of his study, you can see that the pain related to the vaso-occlusive crisis is only, contributes only about 2.5% out of the 100% of the pain episodes ever occurred. What's important to that slide, and those 2.5% relate to the medical utilization. Majority of the pain events occur outside of the hospital and are being either self-treated or not recorded at all. It's also important to mention, looking at that particular diagram, that 45% of the patients having sickle cell disease have not, in that particular study, experienced any pain whatsoever. So what happened as a result of that is that pain, it's clearly is a very extremely subjective parameter, difficult to measure. The second problem is that there is a great deal of difficulty to distinguish chronic pain that results from the accumulation of end organ damage during the progression of a sickle cell disease from the acute pain that can actually be affected by a particular drug treatment. So you are struggling with a relatively narrow window, treatment window, that your drug has to resolve. Now, the first exercise that you can do is you can look at the, let's say, 5 recent drugs that underwent clinical trial, phase 3 clinical trial within the past 10 years. And the interesting pattern that failed and that succeeded. And the interesting commonality between those drugs that did fail, that are listed in that particular slide, is that many of them used change in pain as a primary endpoint. Look at the, those 4 drugs are listed on that particular slide with a reference to the clinicaltrial.gov. And you can, I highlighted with yellow definition of the measurement of pain. It's different, but the commonality between those 4 drugs, they all failed. Another potential approach to develop meaningful clinical endpoint for such disease with such a huge heterogeneity, both in the manifestations and the genotype and the phenotype as well, could be medical utilization. And that certainly was a mainstream for the drugs that did succeed within the past 10 years. Among those is hydroxyurea, L-glutamine, crizanlizumab. Now, the interesting part here is that, is the medical utilization ideal endpoint? Certainly not. And I'll explain that in that particular slide. So imagine that you're trying to, based on this slide that I showed you a few minutes ago, majority of the vaso-occlusive events occur outside of the medical utilization, outside of the hospital. So it would be nice to have a digital health device that would enable you to monitor changes in activity, for example, like it is shown in that particular slide. And what is shown in that slide is the difference of the activity in the activity of the patients without vaso-occlusive crisis and vaso-occlusive crisis. You can see that on the left side of the slide with the red arrow showing the difference. It's important to, and the data came from the Ellipsis trial. It was the observational trial that was run by Pfizer approximately 4 years ago. You can see the difference indicated by red arrow is incredibly narrow. And it only correlates to the difference, at the end of the day, probably within 4 hours before the participants of the study went to sleep. Now, if you look at the right side of the slide, where which is indicated by the yellow arrow, you can see that the difference is much more profound between the medical utilization, where the vaso-occlusive crisis occurred, versus the vaso-occlusive crisis that people experience at home or outside of the medical facility. There could be multiple reasons to explain why hospitalization generates such a profound difference. One explanation that you can come up with is that whether or not you're in pain or not, you wake up in the morning, you have to catch up the bus in order to get to your work. And the actimeter will measure the amount of that movement that you have to endure, whether or not you're experiencing a great deal of pain or not. So the difference will be minimal. Whereas if you're in the medical facility, you're being monitored, you don't have to rush to work, you don't have to catch the bus. Maybe that explains the difference that you can see much more profoundly there. Nonetheless, the point that I'm trying to make of that slide is that, however imperfect the medical utilization could be, whether or not it correlates with the changes in activity or changes in pain in that patient's experience, the primary endpoint using medical utilization certainly works much better. But it's important that the third column from the left is the definition of the vaso-occlusive crisis that is used in those trials that were successful, that all include either the visit to emergency or medical facility or visit to a medical facility in the second trial. So in other words, if you use visit to a medical facility, you can succeed with your drug, however far or however relevant the action of the drug, the mechanism of action of your drug is to the medical utilization itself. All right, so if pain and medical utilization are both imperfect as endpoint candidates, what could be a better one? Let's look at the functional ones, the ones that are not relevant to the vaso-occlusive events. And the first one that is on the list there is change in hemoglobin concentration. And that was the endpoint that was specifically used for the drug that was championed by the company Global Blood Therapeutics in 2018, and the drug name is voxelotor. The idea of the drug is that upon binding to hemoglobin, it restores activity, affinity of the hemoglobin to bind oxygen. As a result of that, there is no polymerization of the hemoglobin occurs, and then the shape of the red blood cells remain the same, and therefore the concentration of the hemoglobin goes up because the hemolysis rates are down. So then the primary outcome was formulated as a number of patients with increase in the hemoglobin concentration beyond 1 gram per deciliter, which is perfect. This becomes a surrogate endpoint that is clinically meaningful and that directly relates to the mechanism of action of the drug. The only problem is that other drugs do not, or sometimes they do not affect the concentration of the hemoglobin itself, so it's not universal. Could there be a universal approach that would be equally efficacious as the hemoglobin concentration? One particular biomarker that has been explored during the past 25 years quite extensively is a hemoglobin F. And hemoglobin F is a modification of the hemoglobin that is being highly expressed in fetus, and the level of expression of hemoglobin F goes down between 6 and 24 months after the baby is born. The difference between hemoglobin F and regular hemoglobin is the affinity to the oxygen of the hemoglobin F is considerably higher, and that could be easily explained. So when the baby is in the womb, it needs all the hemoglobin, all the oxygen that is in short supply. So about 25 years ago, the researcher Martin Steinberg developed the idea that overexpression of the hemoglobin F at the level of approximately 30% could alleviate any manifestation of vaso-occlusive crisis whatsoever. And based on that notion, based on the research that he did, many pharmaceutical companies started developing the approach where they decided not to fix the mutation itself, but instead they decided to overexpress the hemoglobin F to the level that would alleviate or could alleviate all vaso-occlusive events. And that was an approach that companies like Bluebird Bio took. One of the questions that we were facing while interacting with our stakeholders was, what is the threshold? What is the concentration of hemoglobin F that would be could identify ideal patients to be treated with gene therapy? So if you look at the diagram shown on the right side of the slide, you could think that hemoglobin F expression close to 0 will probably do not affect the frequency of vaso-occlusive events. As you move to the right, let's say hemoglobin F expressed in concentration 1% or 5% or 10%, would those patients be suitable for gene therapy treatment? And this is not a simple question, because many of the people have diminished expression of hemoglobin F close to 0, but there are a few expression level of hemoglobin F resides at fairly high concentrations. So for example, some have 10, some have 20, some have 30 and above. You could think of Sherpas who are moving, helping people to get high into the mountains. The expression of hemoglobin F in those people are particularly high. That helps them to cope with the dangerous environment they work in. And that is partially genetic. That could be partially be affected by the level of expression of hemoglobin F, and it varies from different people. So when you're selecting your patients for gene therapy trial, you better be sure what the level of expression of the hemoglobin F is in order to be able to work with the substantial treatment window and be successful with your drug. So when you try to answer that question, and if you take relatively large population of patients, which in our particular case was a cooperative study, which had approximately 3,000 patients with a sickle cell disease, and you try to run the correlation between the hemoglobin F concentration and the frequency of vaso-occlusive crisis, you won't see much of the correlation there whatsoever. You start thinking, what's happening? Why is it not there? Was recently when a group from France came up with the conclusion that it's not just the concentration of the hemoglobin F per se. It is also the number of cells that express hemoglobin F at high enough level. In other words, what they used as a biomarker is the ratio between the concentration of hemoglobin F and percentage of cells, red blood cells, expressing hemoglobin in particular. And the way they did it, which is explained in that particular graph, which I'm not going to walk you through entire graph, but I'll explain it in simple terms. They did stain red blood cells with the antibodies against hemoglobin F. They calculated the number of cells expressing hemoglobin F, and then they also calculated the total concentration of hemoglobin once you lyse all those cells. The ratio between the two give you a much better regression. What is, you can see on the upper left side of the, that particular graph when you compare the correlation between the percentage of the red blood cells versus the hemoglobin F concentration. And that what they did after that, they actually treated the cells extracted from 18 patients with different concentrations of hydroxyurea, and they tried to see whether or not the angle of that regression changes. And they identified relatively narrow window within which that change occurs. The reason I brought you this example is to demonstrate that even when you identify the readout endpoint like that, you can clearly see that that change is not directly proportional. There are multiple mechanisms involved in up and down regulation of the hemoglobin F that may or may not directly correlate with the frequency of the vaso-occlusive events. What do you do then? As a leader of the pharmaceutical company, and I started my presentation saying that there are 2 BLAs are being reviewed by the FDA right now. Both companies, that is Bluebird Bio and Vertex, both coming up, companies that developed the gene therapy treatment, came up with the same solution. They decided to use the probability of the affecting vaso-occlusive events. If you look at that, the upper part is the fragment of the primary, the definition of the outcome, primary outcome endpoint from Bluebird, and the bottom one is the similar one from Vertex. You can see the definition is almost the same. The proportion of subjects who have been affected by the frequency of the vaso-occlusive events. I'm just reading the Vertex one, the one on the bottom. And if you looking at the upper part, one from Bluebird, is the proportion of subjects affected complete reduction of the vaso-occlusive events between 6 and 18 months after the treatment. Keep in mind that both of these treatments are very much similar. Those are gene therapy trials, treatments that have been submitted to FDA right now. So as a summary, the ideal endpoint should measure how patient feels, functions, and survives, and should be meaningful to clinician for having value, be linked to the mechanism of action of the drug. None of the endpoints that are currently available work well for the sickle cell disease due to the heterogeneity of the manifestation and heterogeneity of the disease itself. Changes in frequency of vaso-occlusive crisis are currently used as a primary endpoint of the clinical trial, and the goal of the Sickle Cell Disease Consortium as part of the Critical Path Institute is to identify novel clinical endpoints and biomarkers that could contribute to the improvement of the success in clinical trials in general, and in clinical trial design in particular. Thank you for your attention. Any questions? If not, I'll wait for those questions to arrive later. I was hoping that it could be some parallels between the sickle cell disease and other rare diseases that could be uncovered as a result of that presentation, but maybe it'll take time to uncover those parallels and define those questions better. Thank you.